So moving on to bark, I'd say bark is a little bit less complicated than, or at least the way I want to present it, less complicated than what we saw with the vascular cambium. The vascular cambium, again, produced the wood in the form of secondary xylem, and then also produced secondary phloem in this region. And the other cambium is called the cork cambium, and it's located toward the outside of the plant. And the memorable layer that it makes is the cork or the bark. And then it also produces this phylloderm that we're going to kind of gloss over a little bit. Basically, as the plant grows, you take a little plant, so starting primary stem, and let me outline about the size that it used to be. If it gets out to this size, you can't keep growing and having the same outside, or you're going to end up tearing, right? So if you increase size of something from the inside and you keep one, it's sort of like changing your clothes as you get older. As you were a kid, you got older, you got new clothes. You had to get new sizes of clothes to fit your growing body. So what is the plant's version of getting a new size of clothes? It's got to sort of keep filling in these layers of protection around the outside. For one thing, the epidermis isn't going to cut it. The epidermis was a differentiated single cell layer that was on the outside of the primary growth stem. And as this inside growth increases, that would break apart the epidermis, and that does happen. So the plant needs to replace the epidermis as a protective layer. So it doesn't replace it with more epidermis, but it kind of replaces its function with the function of the bark. So the bark comes in and is now a thicker, more durable, wider protective layer, and it's built by that cork cambium that is able to grow and expand. So being able to expand the protective layer means that it can keep getting wider as the stem gets wider. So we use this term cork, kind of a interesting overlap in the common language of cork with the scientific version of cork. So when we say cork, that's sort of synonymous with bark, but you're probably thinking of things that are more spongy cork. And these are produced, a lot of it from an oak tree. So in the Mediterranean region, there's a tree which is the cork oak. And this was, I don't know what all they were using cork for back in the day, but certainly for bottles to stop up, or sorry, stoppers for wine bottles and so on. This is a spongy type of tissue. It's also waterproof. It's kind of waxy. So it ends up having a few uses: stopping up bottles, spongy soles for your crazy shoes to walk in. And then a little bit of trivia is that cork was the first thing to be observed under a microscope. So these are drawings of sections of cork. Gave us the origin of the term we now use as cells. So it's a little cell, like a prison cell. Somebody said, hey, those look like little rooms. So how is the new bark made? Well, it's made by the cork cambium, and it's produced the bark toward the outside. And the thing is that the bark has to keep filling in. So kind of related again to this drawing, kind of messed it all up with the extra layers of lines in here. But as the growth keeps happening toward the outside, the outer layer splits. It just has to do that from the physics of being forced open from the inside. And the new pieces of bark have to be put in here to anticipate and fill in the gaps that are going to be formed from that splitting. So if it keeps making new cells toward the inside, then there's never an open space for the air to get in, or the water to get out, or the bacteria to get in, or anything like that. So the protective layer is continuous all the way around. Does end up having some layers of extra leftover bark on the outside that are sometimes splitting. So this illustration here shows how the cork cambium has to kind of reset itself toward the inside every once in a while. So this arrows are successive layers, boom, 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 several times that the plant has had to restart making bark from the inside. Sometimes you get wood and you have knots in it. Just an illustration here of the fact that wood, plants as they grow bigger, they're growing past structures that have already been made. And sometimes they have to grow out and around something like a side branch that they've made. So if you get wood and it's got knots in it, those are where the wood of the stem has grown around the wood of a side branch. So these are a little bit less desirable in some pieces of wood. The fancy woods you get, you pay more for not having any knots in it. Sometimes you can get away with them. Sometimes they're cute. Another kind of split in the wood is illustrated here in this structure called lenticels. So they're not really in the center of the image, but what I want to highlight are these little warty things off to the side. These are programmed breaks in the woody stems. And they illustrate the fact that this is a nice, thick, airtight, watertight barrier. But again, we have living cells in there. They need water, as we saw before. They need sugar, and in order to use the sugar, they've got to break it down using cellular respiration, which means they need some air. So trapped inside of the bark are living cells, and they need some air, and that's what these structures do. They're called lenticels, and they let air come in to basically feed the living cells there. And I was always a little skeptical, like they sure don't look like they're very open. But it turns out this graph is showing that they're definitely allowing oxygen to get in. So this is oxygen permeance. And not only does oxygen get in, but the plant actually controls that over the course of a year. So during the growing season, there's more oxygen coming through. During the winter, so through October and then all through the winter, the lenticels are actually not really letting much air in, which is good adaptive strategy because the cells in there are going to be dormant and not needing a lot of oxygen at that time. So the whole plant goes dormant, slow metabolism, not a lot of needs for water or sugar or oxygen, and they kind of close up the lenticels. So they don't look very dynamic to me, but they do have the ability to respond to the environment and change their permeance. Here's a lenticel in cross section, just shows how it kind of looks like a burst open bit. It's not totally open to the environment, but it allows enough oxygen to get through that's adaptive and beneficial. So it's not totally waxed up, as a lot of the bark is a little bit open here. And then basically, as I mentioned, as plants grow, they got to break open their outer layer, which is the bark. And the way that the bark breaks, sometimes it peels, sometimes it goes apart in strips or other patterns. Some people can use these patterns to identify their trees. A good strategy for identifying trees in winter, for example. I'm about 50% on being able to identify my winter, or just to identify a tree by its trunk. Some are very distinctive. The shagbark hickory is unmistakable. This is one of our birches that have this sort of papery bark that peels off like this. I'm going to say this might be one of our black cherries. And then other ones, I even put these images in and I don't remember what they were. So sometimes you can identify them, and maybe somebody would get good enough to be able to identify a tree just by the bark on its trunk. Now I mentioned the living cells that make up the trunk of a tree. They're outside of the wood and inside of the bark. And it turns out if you cut the link between the top of the tree and the bottom of the tree, if you cut it all the way around the full circumference of the plant, you can cut the life stream that allows the water to come up from the roots and the sugar to go down from the leaves. And that effectively cuts off the circulation permanently. So this plant has living cells in its leaves, and they're going to be slowly prevented from getting water. Maybe more importantly, the phloem cells that would transport the leaf water sugars down to the roots, they don't go anymore. So the roots die, the leaves die, the rest of the plant dies. This is a process called girdling. If you want to kill a tree without cutting it down, you can actually do very little to it. Just cut this little strip of damage all the way around the plant, and you can effectively kill a tree that way. And the pattern of where the living cells are, that's also related to the way that trees will grow if they encounter an obstacle. So if a tree needs to grow through a fence or the wire around a tree, the trees do this thing where they kind of bulge out. They can't grow around something like a string of wire. So they end up so they can't grow past it, I should say, or they end up kind of ballooning out around it. And this tree it's going to suffer by not being able to get this living layer any wider than that ring of wire here. So don't trap your trees, let them go free. All right, so here's the whole thing in cross section. We saw the wood, we saw the bark, we saw the narrow layer of living cells. Living cells made up of the vascular cambium, which are alive, the phloem cells are alive, and then beyond that is the cork cambium, those are alive. And as far as what we've talked about today, those are the big layers of living cells. So the cells that make new wood, the cells that make new bark, and the phloem cells are all the living cells in there, and they're all vital to keeping the whole plant alive, really, and especially to keeping the trunk part of the tree alive. And then I'll leave you with this image. I kind of like this is some artist that took a tree apart, and you can take a tree apart down to what it used to make. So this was a very old tree, and the artist carefully took apart the older parts of the tree, and the younger parts are still there. So this is illustrating a little bit of what you get if you get a knot in your wood, is left over of what this little part of tree was grown around to become more trunk. And also illustrating just this general principle of plants growing to fill in around where the cells already were put. So plants don't really get to remove cells once they're laid down. They just have to grow around them.